Hello students, in last lecture we are discussing about Mendelian principles and Mendelian genetics, in which we discuss about monohybrid cross, dihybrid cross, and the three principles of Mendel, which is first one is a law of segregation, law of dominance, and third one is a law of independent assortment. And we also discussed about certain limitations of Mendelian genetics, which is incomplete dominance and codominance. Today we are going to discuss about chromosomal theory of inheritance. There are certain limitations of Mendelian genetics. If we go long time back, when Mendel discovered these things on the basis of their observation in 1816, no one gave them credit for their job, for their work. Mendel ने 1860 में उनके ऑब्जर्वेशंस के आधार पर कुछ लॉस दिए थे और कुछ उन्होंने पेपर पब्लिश किए थे, लेकिन उस टाइम में किसी ने भी उनके ऊपर ज़्यादा फोकस नहीं किया और उन्हें क्रेडिट ज़्यादा नहीं दी. फिर टाइम आगे बढ़ता गया, ऐसे ऐसे 1870 आया, देन 1880, देन 1990, देन 1902. In the beginning of 20th century, two scientists comes up. First one is Sutton and Bovering, who studied the publications and the papers of Mendel and give them credit. और उन्होंने उसके आधार पर उनके मेंडल्स के पेपर के आधार पर एक नई थियोरी की रचना की, जिसे क्रोमोसोमल थियोरी ऑफ इनहेरिटेंस बोलते हैं. तो मेंडल्स की लिमिटेशंस क्या थी? Mendel suggested that each trait transferred by the factors and the factor is present in pair, and these factors segregate during the gamete formation. And then these factors rearrange during fertilization and form a different and new combinations of particular or different traits. So that time people says that yes, look Mendel, you say everything about factors, that traits controlled by factors and factors present in pairs, and they segregate during the gamete formation and rearrange during fertilization. But actually what is the factor? Actually ये factor है क्या? That time Mendel is not capable or not able to explain that what is that factor and where it is present. मतलब जब उन्होंने उससे पूछा, लोगों ने मेंडल से पूछा कि ये आप जो फैक्टर बोल रहे हैं वो असल में फैक्टर है क्या, तब वो एबल नहीं हुआ कि उन्होंने एक्सप्लेन कर सके कि फैक्टर्स क्या है और वो कहाँ पर प्रेजेंट होते हैं. एंड आफ्टर दिस टाइम्स मूव एंड इवोल्यूशन एंड डिस्कवरीज ऑफ मैनी advanced microscope, which leads to discovery of many organelles. For example, structure of nucleus. This is cell and nucleus. Due to discovery of advanced microscope, we know that there are certain chromosomes in every organism present in the nucleus. So on the basis, then Sutton and Bovary said that the factors that Mendel says it is actually chromosome and the genes which is present on the chromosome. So on the basis of these things, this theory is proposed by Sutton and Bovary in 1902. This theory says that the genes are located at specific loci, or we can say locus, on the chromosome, which segregate and independently assort during gamete formation and it rearrange during the fertilization, which means they want to say that the factor that Mendel says is actually chromosome. So this is the two pair of chromosome which is responsible for two traits, one chromosome from parents and one chromosome from male and one chromosome from female. Duplicate and then they segregate. Okay, so the chromosomes is present in a pair and then they segregate during the gamete formation and form haploid gametes, which is said by Mendel in his Principle that the two traits is transferred by two factors. First one is a TT and PP. Okay, let's consider this is two characters and controlled by TT and PP. During gamete formation, what will happen? Four gametes will form T, D, P and P. Okay. So the chromosomal theory is in which Sutton and Bovary says that the factors that Mendel says it is actually chromosome and it is a gene which is segregated during the gamete formation and rearranged during the fertilization. Now let us see the silent features of chromosomal theory. The chromosome presence in pair. As Mendel said, that factor is also present in pair. Second one is chromosome separate during the gamete formation, which is also said by Mendel. Mendel says that factors segregate during gamete formation. In chromosomal theory, Sutton and Bovary says that chromosomes separate during the gamete formation. Third one is fertilization restore the number of chromosome, which means after when gamete form, the gametes have half number of the chromosome than the original parents, which means if let's take one example. In human there are 46 chromosomes, but in gametes 23 chromosomes. In each gametes have 23 chromosomes. After fertilization, both the gametes have 23 and 23. One from father and one from mother will fuse to form zygote, which have 46 chromosomes, which says that the fertilization restore the number of chromosomes. यानी कि fertilization की वजह से chromosomes के numbers restore होते हैं. and the male gametes and female gametes are equally important in the inheritance, which means this is sperm and this is egg. This statement want to say that sperm and egg both are equally important for transmission transfer of trait from parents to offspring. Sperms and eggs both have certain traits, and both are fused to form zygote and new combinations of trait. Here I want to say one thing, that from only nucleus or genetic material is transferred from sperm to the egg, where the egg of female having their cytoplasm too and mitochondria. We all know that in mitochondria there are DNA and DNA is present. So there are also cytoplasmic inheritance. We know that it is transferred from mother to their offspring, which is controlled by the cytoplasm, not the nucleus. So it is only controlled by female. Okay, so this is all about a chromosomal theory of inheritance, which is proposed by Sutton and Boveri. And this want to say that genes are located on specific loci on the chromosome, and the chromosome which is segregated during the gamete formation and restore during the fertilization. So now we will see about the certain another limitations of Mendelian genetics, which is linkage and recombination. So let's see the linkage and recombination. So next topic is linkage and recombination. Like Mendel does his experiment on Pia, the another great scientist Th. Morgan constructed his experiment on Drosophila. In Drosophila, the Morgan constructed many crosses to know the traits which is transferred by the sex gene, which is present in a X chromosome in Drosophila. He crossed between female with the yellow body and white eyes with the male Drosophila having brown body and red eyes. In F1 and in F2 generation, he get some surprisingly result, which is deviated from our dihybrid phenotypic ratio is 9:3:3:1. Morgan says and think this is due to certain physical association of gene on the chromosome, and they discovered the linkage in a Drosophila which is present in a sex chromosome. So let us see what is the linkage. The linkage is when two genes in dihybrid cross were situated on the same chromosome and proportion of parental combination was much higher than the non-parental combination. Morgan attributed this due to physical association of gene and coined the term linkage, which means in dihybrid cross certain genes which is present on same chromosome. Let's take this example as in this example this is chromosome. Okay, let's zoom it. This is a single chromosome which is found in a gametes or sex linked chromosome. This is two gene. First one is for body color, and another one is for eye color. Due to this chromosome present in closely association, these two genes or these two traits will transfer similar combination to the offspring. ये बोथ जीन एक ही क्रोमोसोम पर एक ही जगह पर है, उसकी वजह से उसमें जो कॉम्बिनेशन है वही कॉम्बिनेशन ऑफस्प्रिंग में भी आएगा और उसमें नए रिकॉम्बिनेशन बनने की जो प्रोबेबिलिटी है वो कम हो जाएगी. What will happen in this cross? In this cross, the offspring will have yellow body and white eyes, and another offspring will have brown bodies and red eyes. The combinations which we seen in a Mendelian genetics is not found here because of the linkage. Let's take another example. If one chromosome present here and one chromosome is present here, this is for tall plant and this is for a round seed. What will happen here? Because they are present or far from the another gene, so they will recombine and make new combinations. Where on this hand, in this hand, what will happen? Both the genes are present on a particular locus or particular location on a chromosome. Due to this combination will inherit similar to the offspring. The new combinations will not occur in this process in a linkage. So, linkage reduces the chances of recombination and variation. Now, another term comes is a recombination. So, what is the recombination? Recombination is generation of non-parental gene combination during dihybrid cross is called recombination. We have seen in pea plant in a dihybrid cross done by Mendel, where T12 plant and round cross with the short and wrinkle pea plant. In F2 generation, what we get in F2 generation, we get 4 combination. 2 is similar to the parents. Here, these 2 combinations are similar to parents, but new 2 combinations occur. This is known as a recombination, which means new combinations of non-parental genes occur. So this is called a recombination. Here, the linkage and recombination are to both the opposite word. Linkage reduce the variation in organism or in a particular organism, where on the other hand recombination increases the chances of variation. The recombination will give chance to the organism to evolve, and linkage will decreases the chances of evolution in a particular organism or in a particular species. So this is about linkage and recombination. Let's summarize what is the linkage and recombination. Linkage means the genes which is present on a particular chromosome will inherit as in a parental which is present in a parental combination, which means जो parental combination है वही offspring में ट्रांसफर होगा. इसे हम लिंकेज बोलते हैं, विच इज ड्यू टू फिजिकल एसोसिएशन ऑफ जीन, एंड रिकॉम्बिनेशन इज द जनरेशन ऑफ नॉन पेरेंटल कॉम्बिनेशन known as a recombination, which gives a chance to variation, which gives chance to the evolve, to species to evolve. So this is about linkage and recombination. Now next topic is sex determination in organism. So let us see sex determination in different organism. Now next topic is sex determination. Now what is sex determination? My dear students, as we all know that one sperm and one egg cell fuse to form zygote. Okay, but the question is, the zygote will form into male or female. How it will decide that this zygote will go and form into a male or female body or female organism? So this, to determine this whether this zygote will form into a male or female, is known as sex determination. First of all, the Hanking in 1891 observed a specific nuclear structure found in certain sperms of certain insects, which means the Hanking found certain specific structure which is found in a sperm of particular insect. So consider this is the sperm. He found a particular structure in only 50% organism, 50% sperm, and found absent in a 50% sperm of that insect. So, but he is not able to explain that what is the role of this specific nuclear structure. Later scientists decide that this specific structure is responsible for formation of female, and this structure known as later this structure considered as a X chromosome. Now the sex determination is mainly divided into two groups. First one is a male heterogamy and another one is a female heterogamy. In male heterogamy, the two types of gamete will form by the male, which means in male जितने भी स्पर्म्स प्रोड्यूस होंगे वो दो तरह के होंगे. फॉर एग्जाम्पल इन ह्यूमन, %50 sperm having X chromosome and 50% gametes or sperm having Y chromosome. Likewise, in Drosophila also, the 50% sperms have X chromosome and 50% gametes or sperm don't have X chromosome. So this is called male heterogamy, which means the male produce two different kinds of sperms, known as male heterogamy. On other hand, female heterogamy is also similar to the male heterogamy, but in female heterogamy, female will produce two different kinds of gametes or eggs, we can say, and male will produce two similar kind of sperms or gametes. So this is the male heterogamy and female heterogamy. Now we will discuss about particularly human sex determination, that how sex determined in human. In human, we all know that 23 chromosomes are there. 23 chromosomes are there. Out of 23, 22 chromosomes are similar. And last chromosome is different in male and female. As we all know that in last chromosome, in male, sex chromosome will have X and Y, where in female it will produce only similar kind of X, having X chromosome. Now what will happen in male? Male will produce two types of sperm, we can say, also called as a gamete, and in female will produce a similar kind of eggs. Let's see the cross between them, what will happen. Here, if this sperm fuse with the eggs having X chromosome, what will happen? Here, female will form, the zygote will grow into female, where here the sperms having Y chromosome will fuse with the chromosome with the eggs having X chromosome. This zygote will grow into male organism. Here also XX will form into a female and XY will grow into male. Now we can calculate that the chances of male and female is 50%. Now questions come arise that अगर यहाँ पर हम देखें systematically तो there is chance that 50% birth of babies or new children will male or 50% are female. But we see in our community, or we see in our state and our country that this ratio are not followed. In our state there is a 100 female and 973 what 900 something females are there, which means this ratio is not followed actually. So now you have to answer, or you have to find that why this ratio is not followed in actually. So production of male or female organism is due to a male organism in human, which is not controlled by female organism. Now mutation. What is the mutation? Mutation is defined as a certain change in a DNA sequence which result into a different in genotypic and phenotype known as a mutation. There are different kinds of mutation. First one is a point mutation, where only one base pair is changed, and another is a chromosomal mutation, where number of chromosomes are different. So in certain organism the chromosomes number will increases, or in certain cases the chromosome number will decreases. So this is the XX. But in some organism, these eggs don't segregate during the gamete formation, and it transfer similar in the gametes. When this gamete will fuse with the another X chromosome, it will lead to produce 3. The female having 3 more, one more chromosome, one more X chromosome, which is known as a super female. Okay, and the characters of this female will change from the normal female. The mutation is occurred due to certain natural processes like linkage and other processes, and it also occurred due to some environmental condition or certain mutagens. Mutagens is the chemical or any chemical which produce mutation in a particular organism known as a mutagen. Mutation is also occurred by UV and X-ray. We all know that. In a point mutation, only one base pair will change. A change in only one base pair lead to certain formation of disease. Like in human, we all know that about sickle cell anemia. This is occurred due to change in only one base pair. So this is known as a point mutation, and this is about mutation. Now pedigree analysis. Now next topic is pedigree analysis. What is pedigree analysis? Pedigree analysis is defined as analysis of trait in several generations of particular family, which means जो कैरेक्टर्स और ट्रेड ट्रांसफर हो रहे हैं जनरेशन टू जनरेशन, उसको हम ज़्यादा जनरेशन में स्टडी करते हैं तो उसको हम बोलेंगे पेडिग्री एनालिसिस, विच इज़ यूज्ड फॉर टू ट्रेस अ पर्टिकुलर ट्रेड और पर्टिकुलर कैरेक्टर्स. मतलब हम उससे पेडिग्री एनालिसिस से पर्टिकुलर ट्रेड और पर्टिकुलर कैरेक्टर को हम ट्रेस कर सकते हैं कि वहाँ वो कैसे रिएक्ट करेगा और कैसे वो किस ऑर्गेनिज्म में प्रोड्यूस होगा या ऑफस्प्रिंग में प्रोड्यूस होगा या नहीं होगा. So in pedigree analysis, this symbol is for female and this symbol is for male, and here is link, that means they are getting married. And this is the symbol of that female having affected with the particular trait, and this shows female is not affected with the particular trait. It will form a offspring male, female and male. Then this offspring, out of 3, one male have affected with the trait having their parents, which married with the female, which also have that, which is also affected by the particular trait. Though on the basis of that we can predict that this trait will, there is lots of chance for this trait to transfer in a particular or individual's offspring. Okay, so this is known as a pedigree analysis. So here we already discussed about sex determination, mutation and pedigree analysis. Now in next class we will discuss about certain genetic disorders in a human. Thank you.